what we tend to do is use sequences from other genomes to match against the human genome to work out where the proteins are. That was the main thing we did, was asking where the proteins are. At the time, for a whole bunch of different reasons, people thought that we had 100,000 protein-coding genes in the human genome. And there are lots of reasons why that estimate had been made, but it was an estimate made on very, very little data. And our very first runs, where we tried to decode the human genome, we came out with far, far fewer genes than we should have. Just tiny amounts, between 20,000 to 30,000. And to be honest, we didn't believe ourselves, nor did the people around us. And so during that very first analysis, we went through a process to try and work out where we'd gone wrong and estimated a number that was around 35,000 protein-coding genes back in 2000. And that was an estimate based on the data because the human genome was incomplete. Now, in fact, in retrospect, our very first estimates were probably more on the money. It now is reasonably clear that there is probably just about 20,000 protein-coding genes in the human genome. This is far fewer, a fifth of what we originally thought when we started the process. And it's the same number, for example, as a worm, a small worm that lives in the soil.